My name is Junxiang and I'm going to walk you through my poster named Spatial Multimodal Profiling Rivals Spatially Structured Gene Program in Tumor Microenvironment of Non-Functional Pancreatic Neuroendocrine Tumor. So, pancreatic neuroendocrine tumor, or PANNET, is the second most common malignancy in pancreas, though the incidence and prevalence of these tumors continues to rise globally. In order to get good prognostic markers, a lot of studies has been carried out over the past few decades. One of them is ALT, or alternative lengthening of telomeres. Studies has shown that the activation of this ALT mechanism has shown a remarkably decrease of survival rate compared to alt-negative rates. In addition, several transcription factors such as ARX, PDX1 has shown their abilities to regulate the islet cell differentiation, which is actually where the PANNET originated from. However, these biomarkers are characterized in bulk, and PANNET, just like all other tumors, is a very complex disease and has a lot of heterogeneity. So far, very little is known about the tumor microenvironment as well as the spatial organization within the tumor cells alone. So, our objective is that we want to comprehensively profile PANNETs by integrating spatial multi-omics together with already established biomarkers to define subgroups of PANNETs and explore the underlying biology underneath. So here goes the study design. We integrated 62 patients in total to generate two tissue microarrays with a balanced cohort of alt-negative, alt-positive, and a few other biomarkers. And after the tissue was generated, it was sectioned onto different slides, and we run the IHC staining with the well-known biomarkers. And on their adjacent slide, we run a spatial barcoded sequencing platform called the VZM cytosist from 10x Genomics to unbiased characterize the spatial transcriptomics. And on the adjacent slide, we use the spatial proteomics platform with this sequential staining of IF from the Lunafo platform. And together, we have a multimodal and multi-scale data set. So we first take a look at this unbiased clustering just based on the gene expression profile from the VZM dataset alone. So on the top left, this is the 5 experiment of the covered TMA course, and on the right is actually a UMAP calculated based on the PCA space of a VZM spouse, simply on the gene expression. And in this image, we can actually see that these spots are manually annotated by the pathology annotation, including normal arsenite structure, normal ductal structure, normal islet structure, stromal region, immune-rich region, and a few other tumor subtypes from the pathology annotation. And on the right is actually the clustering result from the gene expression space. We then first run a quick differential gene expression analysis in the alt-positive versus the alt-negative tumors. And here is the volcano plot, shows the differential expressed genes. And among the ALT-positive genes, the top hit of the hallmarks of cancer shows that the oxidative phosphorylation is activated in ALT-positive tumor, and in contrast, in ALT-negative tumor, the NF-kappa B pathway was activated, which kind of explains the survival difference. And also, in addition, compared to the bulk data, we actually didn't see a lot of EMT and ECM remodeling in the ALT-positive tumors. We next use the top markers of a reference single-cell RNA dataset to generate the epithelial, stromal, immune, and malignancy signature, and calculate a gene set enrichment score across all VCM spots. And we compare this signature scores with the pathology annotation we get from figure 3. And then we recalculate the gene expression UMAP, but this time only use the spatial co-expression modules, not the PCA scores. And by plotting the signature score on this UMAP, you can actually see that all the normal cells are clustered together, showing this epithelial signature. Stromal is a little bit sparse, but still overall integrated all together. Immune is concentrated in one small subset of all VCM spots, and malignant seems to have a lot of distinct patterns of organization just based on this UMAP. And then we dig deeper into these spatially co-expression modules. What does it really mean? We used this example patient to visualize the spatially organized gene modules. So notably, this is actually a tumor center, and this in the middle is normal arsenine. So in this, when plotting this gene program spatially, you can see that this gene program is highly expressed in the regions that is in close contact with the normal region, but sort of less expressed in the tumor center. And this module is the opposite. It only expressed in the center of the tumor, but not expressed in the regions that is close contact with the normal arsenine. And we can also expand these type of observations into multiple patients. We can just simply characterizing the spatial co-expression modules for across each patient, and then cluster all the modules together. And we can see that there are a lot of potentially shareable programs across different patients. We then look into the Lunaphore data, which is a different modality, including the protein data. We first run unbiased latent clustering, which is shown here. Notably, we also see this tertiary lymphoid structure here. This is a confirmed tertiary lymphoid structure by the pathologist, and this is the protein marker expression within this region. And we can then next pull out a single cell expression data set in this example core and label that into different cell types. And we can use spatial distance to map the spatial connection between different cell types. In addition, we can also identify niche within a certain region composed of different cell types of this example core. So to sum up, we can draw a few little conclusions is that first of all, the batch effect is minimal. The information from different modalities is also share concordance, and we have some initial findings about all positive versus all negative, and we'll keep exploring the spatially organized gene programs and the spatial niche compartment, and try to integrate the latest deep learning based algorithms to learn from all the modalities.